Alright, I'm going to go ahead and start glycolysis here. Um, Alright, here we go. So, glycolysis. Alright, so this is the breakdown of glucose. So we got that done. What I want to emphasize is that this is in the cytosol, or the liquid of the cell. So, you know that you're going to have glycolysis, you're going to have the Krebs cycle, and you're going to have the electron transport chain. The glycolysis will occur in the cytosol of the cell, and the Krebs cycle and electron transport chain will occur in the matrix of the mitochondria. Um, so I just want you to make sure you know that. So here's what glycolysis is going to do. You're going to start off with glucose, which is a 6-carbon molecule. You are going to convert this to G3P, which will be converted to pGal, familiar from photosynthesis. From pGal, it's turned into BPG. So BP is biphosphate. Biphosphate. So here what happens is you actually add a phosphate group. And eventually the BPG is turned into pyruvate. This molecule is very important because this is the end product of glycolysis. Pyruvate will then be used to either go into fermentation, which I'll go on later in this video, and pyruvate will be used for the Krebs cycle if you have an aerobic respirating organism. So what I want to also emphasize in glycolysis is this is energy investment. So, the stock market, if you're going to try to make some money, you're going to put some money in to get some money back, right? So here, what you're going to do is you are going to put in 2 ATP in the beginning. And what you're going to do is you're going to have 4 ATP out. So your net ATP equals 2, because it's 4 minus 2. Sorry, this should be products right here. Products. Net ATP 2, 2 NADH, and you're going to have 2 pyruvate. So if you have trouble remembering pyruvate, here's what I want you to do. So glucose is a 6-carbon molecule. It's going to be broken down to 2 3-carbon molecules called pyruvate. So this is glucose, and these are pyruvates. Okay. So let's go ahead and make a new sheet. All right, so we have now gotten to the Krebs cycle. All right, so here we go, Krebs. All right, so the Krebs cycle is going to take place in the mitochondria. And this is also called the citric acid cycle. TCA cycle. So here's what happens. So in the Krebs cycle, your pyruvate is converted to acetyl-CoA, which is a 2-carbon molecule. This will then enter the Krebs cycle, and here's what's going to happen. So you are going to produce, for every 1 pyruvate, you will get 3 NADH, 1 ATP, 1 FA, uh, 1 FADH2, and you will also, here's the thing, so you also produce 3 CO2. What I also want to highlight is when you look at the cell resp formula, your reagent is glucose plus O2 yields, this should be a 6 CO2 plus 6 H2O. So look at this. If you recall, glucose produced 2 pyruvates, right? So we've got 1 pyruvate, which produces 3 NADH, 1 ATP, 1 FADH, and 3 CO2. That means in order for 2 pyruvates, which is actually equal to 1 glucose, because glucose is a 6-carbon molecule and pyruvate's 3 carbon. So, if you follow the atom conservation rule, you got 6 carbons and 2 times 3, 6 carbons. You're going to get 6 NADH, 2 ATP, 2 FADH2, and 6 CO2. So watch, this 6 CO2 right here corresponds to that part of the formula. So you really want to know that 1 glucose will produce, let me do the math for you right here. Sorry. So if we add up all the stuff from glycolysis and the Krebs cycle, let me just write this down for you. Products. I'm actually only putting the energy products here. So for 1 pyruvate, here's what we're going to get. 4 ATP, 5 NADH. Oh, sorry, actually this is not for 1 pyruvate, this is for 2 pyruvates, aka 1 glucose. 4 ATP, 5 NADH, 2 FADH2s. Also, what I also want to mention to you is that in the Krebs cycle itself, you'll have 2 carbons leaving. And this third CO2, this is actually taken out when pyruvate is converted to acetyl-CoA. Because watch, so pyruvate is 3 carbons, acetyl-CoA is 2 carbons. You had to lose a CO2, you had to lose a carbon somewhere, so this C actually leaves in the form of CO2, and once the rest of the Krebs cycle occurs, you will leave with 2 CO2. Okay, so let me move on to the electron transport chain. Okay, so here's basically the whole idea of the electron transport chain. So your energy molecules NADH and FADH2 will donate H pluses. So let me draw out a cell membrane for you. And I'm going to put a little motor here. This right here is ATP synthase. So you're going to have your FADH2 and you'll have NADH. And what they'll do is they will transfer H pluses to this side of the cell, right? So these are gone, and this becomes FAD, FAD, and NAD plus. So these become oxidized, and they'll lose their hydrogens. So here's the deal, though. If you recall back to diffusion, diffusion is a flow from high concentration to low concentration. So these H pluses will move through ATP synthase, and essentially this is like a if you've ever seen a water wheel, the water moves the wheel. In this case, the H pluses will move the ATP synthase, and when it does, ATP synthase will have ADP plus a free organic phosphate to form ATP. So this is basically the reason why the electron transport chain produces so many ATPs, is because here you've got the oxidative phosphorylation, and your H pluses are providing the chemical power to, or the chemical work to produce ATP. Now here's something I also want to emphasize. So if you recall, H plus will lead to acidic environments, but down here you actually have an oxygen as an acceptor. This is where glucose plus O2 comes in, because O2 is your acceptor. So when you have H plus O2, you will form 2H2O. So this is where cell respiration will produce the water. So this is where all the water is coming out when you're breathing. Um, this whole process of uh, H plus powering is called chemiosmosis. So if you remember, osmosis is basically the movement of water. This chemiosmosis is a movement of chemicals. In this case, it's going to be H plus. So O2 equals final acceptor. And the O2 will lead to neutral H2O, aka good old water. And you're not going to have acidic environment in your cell, which could lead to protein shape degradation or denaturing. The last thing I want to do is fermentation. Fermentation, sorry. Don't know why I can't spell today. All right, so there's two types. So one is going to be alcohol, and two will be lactic acid. All right, I'm going to move this down. Okay, so in both cases, you will have a pyruvate plus NADH. And here's the thing, it depends on your product. So here you're going to have lactic acid plus NAD plus. You're going to produce ATP, but you're only going to produce about 2 ATP. You're not, it's very, very inefficient. Inefficient. Now, you may ask, oh, and here it's going to be pyruvate plus NADH to form alk plus CO2. This is the other thing. Alcohol will be you'll have CO2. Lactic acid, you will not produce CO2. Also, one last thing I want to highlight here is that this is in yeast. So bakers, we use baker's yeast, and we've been using yeast to produce alcohol, but we also use yeast to create the fluffiness in the bread, and the fluffiness is all coming from CO2. Lactic acid. So when you work out, because this is no O2, sorry, I should have said this earlier. Fermentation is going to be no O2. Because you don't have oxygen, because you're not breathing very constantly when you're working out or like sprinting, you're going to produce lactic acid. And when you get sores and cramps in your muscles, this is essentially what's going on. It's a buildup of the lactic acid in your muscles, which cause cramps. Not fun. No one likes cramps. You just take Advil if you got cramps, or you can have bananas. So you may ask, why do you need O2? So sometimes, or why do you not need O2? So here's the thing. It's a little outside of the course, but some bacteria, such as you've got bacteria. Yeast is actually a fungus. They can't use oxygen in the Krebs cycle and the electron transport chain. Recall that in the ETC, O2 is final acceptor. These cannot use O2. So no O2. This is really important because if they use O2, they could become